tomorrow. Other than that, let's get started. I want to introduce the brand new Dean of the College of City College, New York, and BMES President, Gilda Barabino, to introduce the Pritzker Distinguished Lecturer this morning. Gilda.
Good morning, and welcome to this plenary session for the Pritzker Distinguished Lecturer Award, the highest award bestowed by the Biomedical Engineering Society. The Pritzker Distinguished Lecturer Award is presented each year to an individual to recognize outstanding achievements in leadership, in science, and the practice of biomedical engineering. This year's winner is Dr. Chilkoti. Dr. Chilkoti is the Theo Pilkington Professor of Biomedical Engineering at Duke University. He has secondary appointments in mechanical engineering and material science and chemistry. He's also the director of the Center for Biologically Inspired Materials and Material Systems. He received his Ph.D. in chemical engineering from the University of Washington, and he's been a faculty member at Duke University since 1996. His bioengineering research explores the interaction between biological and synthetic polymers with biology at the molecular level, with the goal of developing molecular tools and devices for medicine and biotechnology. His work in biomolecular engineering focuses on genetically engineered stimulus-responsive biopolymers as tools for protein separation, bioconjugation, and drug delivery. He has co-authored over 250 publications and has more than 70 patents and patent applications. He's the founder of 2 startup companies. His first, Phase Biopharmaceuticals, has raised $65 million in venture capital funding and has taken drug delivery technology that he developed into Phase 2 and Phase 1 clinical trials for 3 drugs. A second, more recent venture, Scintillas Incorporation, is commercializing a point-of-care diagnostic based on a polymer brush technology developed in his laboratory. He received the Career Award from the National Science Foundation in 1998, the 3M Non-Tenured Faculty Award in 2002, and the Distinguished Research Award from the Pratt School of Engineering at Duke in 2003 and 2005. He was a recipient of a Senior Researcher Award from the Alexander von Humboldt Foundation in 2010, and the Clemson Award for contributions to the literature from the Society for Biomaterials in 2011. He was elected Fellow of the Controlled Release Society and the Biomedical Engineering Society in 2013. He also serves on the editorial board of 5 journals. His talk today, Solving Drug Delivery Problems by Genetically Engineered Polypeptides, focuses on the power and versatility of genetic engineering to develop molecularly engineered drug delivery systems. His talk will highlight 2 orthogonal designs of genetically encoded peptide polymers, nanoparticles and gels, for drug delivery in 2 different therapeutic arenas, cancer and type 2 diabetes. It's with great pleasure that I present the 2013 Pritzker Distinguished Award Lecture to Dr. Chilkoti.
This is probably the largest crowd I've ever had. So I have to say at the outset that I'm actually honored and sort of deeply humbled by this award. I was extraordinarily surprised when I got it. I wasn't paying much attention to the fact that I'd been nominated because I was convinced I wouldn't get it. So thank you to the unnamed selection committee that decided to give me this honor, and thank you all for being here. So today I'm going to sort of take you through sort of work in my lab over the last 10, 15 years, as was described in 2 different areas. We worked in cancer for a long time, and more recently type 2 diabetes, and different designs that we have developed using polymers where we write out sequences on paper, make genes, and then use E. coli to make us the polymer of interest. So I'm going to start out with a very simple proposition that I think most of us in the drug delivery field would agree is true, that despite trillions, if you look at it historically, all the money that has gone into developing drugs, most of it actually goes into developing the drug, and drug companies hardly, if ever, think about how to deliver them. If they make it into a pill, who cares if it's insoluble? It'll get there somehow. Let's inject it. If it's not very soluble, just add some toxic chemophore and let's live with it. So if you look at all the different classes of drugs, small molecules and then peptides and proteins are sort of one broad class, and nucleic acids, they all have extraordinary problems. In fact, with silencing RNA, as Philip Sharp said, there's only one problem with silencing RNA, drug delivery, is the delivery problem, right? I'm not going to talk about that today. I'm going to focus on two other classes, small molecules, because they still represent probably the largest number of drugs are small molecules. And I'm going to talk about small molecules in the context of delivering chemotherapeutics for cancer. And then I'll switch gears in the second part of my talk and talk about peptide drugs, because there are about 650 in the preclinical and clinical pipeline, and they all have some pretty serious problems, even though they are quite potent drugs, because they're often derived from naturally occurring peptides in the body. If you look at small molecule drugs, I had a very brave student, Sarah McEwen, who went into the literature and actually dug up the molecular weights of a lot of different drugs, and this is what we find, and this is something we knew, but we wanted to quantify it ourselves. This is sort of one example of Lipinski's rules, that drugs are designed to be 500 molecular weight or less. This is sort of a kind of almost Gaussian distribution you get. So you have a molecule that's small, and you're going to take it, if you inject it, it's a small molecule, it's going to be cleared to the kidney very quickly. That is, if you're lucky, if it's soluble, okay? And so if it's small, one trick to improve its distribution within the body is to make it larger. And that's the whole area of attaching it to polymers and so on, or putting it in nanoparticle, which increases the size. But the second problem is, especially in cancer chemotherapeutics, is that they're hydrophobic. They're called brick dust in the parlance. So you have a molecule that's very small, that doesn't want to go into solution to begin with, and you're trying to cure a deadly disease like cancer. So here again, Sarah went in and plotted something called a log D, which most of you would know from physical chemistry is the octanol water distribution coefficient. So it's simply how hydrophobic a molecule is. So things that are over here are very hydrophobic, these are hydrophilic, and you can see there's a strong bias towards drugs that are very hydrophobic. So for example, 4 is brick dust. It has absolutely no solubility. Even 1.5 is quite hydrophobic. So this is the second problem, that these molecules are hydrophobic and they're small. And so the second problem that has to be addressed, even before you think about injecting it in, is you have to solubilize it. It has to be soluble. And then the third thing is, when they go in the body, they have suboptimal distribution, and this is where nanotechnology has turned out to be quite handy, because it turns out by changing the size and shape of the delivery vehicle, especially at the nanoscale, you can do some pretty powerful things in controlling the fate and disposition of drugs within the body. Now when you take a drug and you sort of formulate it even before it goes in the body, once it goes in the body, this is an enormous problem, because if you think about the size scale of a protein or a nanoparticle as it traverses from its site of injection to its site of action, it's actually the same relative distance that it would take for a rocket to go from here to the moon. So the length scale is about the same. This is even if there were no other barriers. So you inject it and then you traverse this enormous distance. And this distance that you traverse is not an easy distance to traverse, in the sense that there are many, many barriers faced by this drug or this delivery system on its way potentially from its injection site or site of administration to the site of action, which is typically inside the cell somewhere. First thing that's going to happen is it's going to get cleared, and the body has exquisite ways of clearing things through the kidney and the RES system. And then, if you're trying to deliver a drug to the tumor, there are a huge number of barriers at different length scales, which first started with the fact that there's a convection gradient in tumors, but it's the opposite of what you want. So you're fighting against a pressure that is higher at the core and lower at the outside. And then this drug has to sort of hack its way through almost like an Amazon jungle, like a stroma-rich environment. I sort of think about this drug as an explorer in the Amazon jungle. It's sort of, the rivers are flowing in the wrong direction, and it has a little machete, and it's trying to hack its way through this jungle, which is rich in ECM. And then it reaches a cell membrane. Think of it as a fort and a moat, and the moat is a cell membrane, and you have to traverse it, and many drugs will not, especially hydrophilic drugs. And then once it goes into it, the drug gets sort of thrown into this vat of acid, which is endosomes and lysosomes, and it's trapped there and degraded, and it has to quickly make its way out, otherwise it won't work. So you can sort of see this enormous problem that a poor drug or a drug delivery vehicle faces when it's trying to get to the site of its action somewhere within the cell, either in the cytoplasm or the mitochondria or the nucleus. So there are, of course, many, many delivery systems that have been developed. And if you go through the literature, you'll find a dizzying array of systems. And they all have their strengths, and I would argue they all have their weaknesses. So you can attach drugs covalently to make them bigger. So there's an example of an antibody attached to a polymer, or a peptide attached to a polymer, or an antibody attached to a small molecule drug. So drugs can be attached covalently to carriers to confer desirable properties. Drugs can be physically encapsulated in things like liposomes or micelles. And you can also electrostatically complex drugs onto larger entities. So my argument is that if you look at these carefully, many of them are quite useful, and some of them have made it to the clinic, but few of, or if any of these meet a lot of the requirements that you want in a drug delivery system. And what I want to talk about are elastin-like polypeptides today. These are polymers that we've been working with for a long time, which some of the time have many of the properties that you look for. Not always. They're not perfect. No system is perfect. But these do have some useful attributes. Elastin-like polypeptides are peptide polymers where the repeat unit is just a 5 amino acid sequence stolen from biology, stolen from a protein called tropoelastin, which is the repeat unit, from tropoelastin. So this 5 amino acid sequence is found in tropoelastin. And what's going on here? Sorry. And these have very interesting behavior, that they have lower critical solution temperature transition behavior, which is shown here. As you raise the temperature, they go through a phase transition, they become insoluble. And when you lower the temperature again, they become soluble. So these are sort of very simple, smart polymers where they have a physical trigger. You raise the temperature, they change the physical state, and I'll show you some examples of how you can use this change in physical state to play some games in drug delivery. What is nice about the system is that this temperature at which this transition occurs can be tuned very precisely by control of two orthogonal variables. And the two variables are the chain length of the polymer and the molecular weight. And because we make these polymers using molecular biology, we have complete control over these two orthogonal variables, so we can design an elastin-like polypeptide at will, so to speak, with a transition temperature with the precision of a few degrees Celsius for any given application. So these are made, even though these are polymers, where the repeat unit is 5 amino acids, they're made using genetic engineering. And so we have developed ways to assemble genes very rapidly and precisely, because the problem in molecular biology is that if you want to make a long polymer, you have to start with the DNA sequence, and you cannot make very long DNA sequences. So you make short DNA sequences, and then you have to assemble them. And we have developed in the last decade many different methods to assemble genes to the right length. We take these genes, we put them in a vector, into a plasmid. Then they go into a cell, into E. coli. Then you do a typical fermentation, just like you would brew beer. And the elastin-like polypeptide hopefully will be produced at a high concentration within the cell. You break the cell open. The polymer in almost all cases is soluble, and then we have developed a purification method that can be scaled up very easily that takes advantage of the phase transition behavior. So we can purify grams of this material in the lab without doing any chromatography, and those of you who've done chromatography know that it's tough enough to do 10, 20 milligrams, and when you think about gram scale, it gets very expensive and very tedious. So we have at this point a fairly seamless way of designing genes, putting them into vectors, putting them in cells, and producing things, and this can be done in a matter of weeks. So why ELPs for drug delivery? Because I've said there are a lot of different systems, and some of them have made it to the clinic, so I feel like I should put up one slide to sort of show you why we think this is a useful system to work with. First of all, because these are smart polymers that can be tuned precisely at the molecular level. Because we are going to write a gene and then use the proofreading machinery of the cell to make a precisely defined polymer sequence, this is where they differ in a significant way from all synthetic polymers. These are completely monodisperse polymers where we have complete control of the sequence at the molecular scale. So we can control the molecular weight precisely, and they're completely monodispersed as a consequence of being genetically encoded, and they're main chain biodegradable. When you inject them in the body, even the most non-degradable system we've made degrades at the rate of a few % a day. So these things are going to be degraded, which is good because ultimately, for chronic diseases especially, you don't want your delivery system to accumulate in the body. And the breakdown products are non-toxic. We can produce them at high yields, and we are lucky here because there are other peptide polymers derived from silk and collagen that cannot be produced in bacterial expression systems at high level. So just simply luck in a sense, we can produce a lot of it in shaker flask culture, so you can even make gram scale amounts for scaffolds and so on. And the fact that they have an LCST trigger, the fact that their solubility can be so easily turned on and off with temperature or by the addition of salts means that we have a very simple way to purify it. And we can also use the LCST trigger to design systems that do something interesting. So for example, this is an example of a diblock copolymer, an elastin-like polypeptide, which is completely, where its assembly or controlled by pH in a very narrow temperature range. So these micelles are micelles at 7.4, but if you go down to the tumor pH of about 6.4, they dissolve, and if you raise the temperature again, they form again. So this is an example of a system that is both temperature and pH responsive in a very narrow temperature range and pH range that is useful for biology and medicine. I won't talk about these more elaborate systems because I actually want to share with you sort of a story that is more translational, where we've designed the first generation systems and taken them all the way through multiple animal models. But I just wanted to show that you can build fairly elaborate and fancy systems that respond to very narrow changes in the environmental conditions. Elastomeric polypeptides, I hope to show you today, are enormously flexible, that many, many different strategies for drug delivery are possible. And in the context of cancer, I'm going to start out with what I would call the simplest, stupidest way of using them is to simply make a polymer that is long so that it doesn't get excreted very quickly, and very hydrophilic, so with a very high transition temperature, because what we want to do is we want to use this very long hydrophilic polymer, and we want to attach hydrophobic drugs to it to do two things: to improve the solubility and to make them be cleared less quickly through the kidney. And then the second thing we want to take advantage of is the fact that if you look at the tumor vasculature, it's leaky. So normal vasculature is not leaky, but tumor vasculature is leaky. So if you can keep a high enough concentration of a molecule, such as a hydrophilic macromolecule with a drug attached to it, for long enough in systemic circulation, you can use diffusion as a driving force to drive your drug attached to your polymer out into the tumor stroma. So this is called passive targeting, and another term for it is the enhanced permeability and retention effect. The other side to this equation is also the fact that the lymphatic drainage system in tumors is poorly developed. So things can accumulate passively by diffusion, and then they're not cleared very quickly. And the flip side of that, unfortunately, is that the tumor pressure builds up, and you have this interstitial fluid pressure, which makes convection very difficult to use as a way to deliver drugs into tumors. So I'm going to first talk about a hydrophilic polymer, about 60,000 molecular weight, and we're going to attach some chemotherapeutics and see what happens. That's the first part, little vignette I want to share. And then I'm going to show you a sort of an expansion of that idea, where we then take the system and make it thermally sensitive in a very narrow temperature range between 37 and 42 degrees Celsius. And you might ask, why 42? Why this arbitrary number? It's because you can heat patients up to 42 degrees Celsius. You can even actually do whole body hyperthermia up to 42 for a couple of hours. You can get clinical protocols approved, and it's actually been done clinically as an adjunct to external beam radiotherapy. So 42 is just enough to change the physiology of cells, but not enough to destroy them or cause necrosis. And then finally, I'm going to talk about the idea of taking elastomeric polypeptide that has a body temperature that's lower than body temperature, and then creating an injectable hydrogel-like material out of it for systemic delivery of a peptide drug. Before I go any forward, I had to sort of disclose that all this technology has been licensed to Phase Bio, but nothing I'm going to show you today comes from the company. So I believe in separation of church and state, so to speak. So they do their thing and we do our thing. So this is just academic research, but hopefully of some practical value. So we've been working on this problem of attaching small molecule drugs, hydrophobic drugs to elastomeric polypeptides, and Jonathan McDaniel, who's here, took on this really brave task of figuring out what happens when you attach small molecules to these kinds of elastomeric polypeptides, which are long, about 60,000 molecular weight. So here's the polymer that he designed for these drug delivery experiments, which is a long BPG-XG sequence, 160 pentapeptides. Okay, so this is many, many hundred amino acids long, with a very, very short drug attachment domain. So it's just cysteines separated by a couple of glycine spacers simply to space out the reactive sites, and we're going to take hydrophobic drugs, attach them to the thiol of the cysteines, right? So this is the reactive site. The rest of the polymer has no reactive sites. And the idea is we're going to attach these creating almost like a boat anchor. And what he found was that when he took different molecules and attached them to this polymer chain, if the molecules were sufficiently hydrophobic, they would self-assemble. But what was even more interesting was the fact that he found a very simple correlation between the hydrophobicity of the molecule and the propensity to self-assemble. So if you look at all these molecules here, these are fairly reasonably hydrophilic, going on around the circle of hydrophobicity to brick dust, things that are absolutely insoluble. Molecules shown in sort of this blue or purple here, if you attach them, they do not trigger self-assembly. Anything with a hydrophobicity with a log D greater than 1.5 self-assembles, and when it self-assembles, it makes spherical nanoparticles. More precisely, it makes things that look like star-like micelles. There's a small core where the drug is covalently attached and sequestered, and then these arms extending out. And you can see this in these cryo-EM images that were taken by Kevin Wargo and Dan Hammer's group at UPenn. If you have a log D of 0.9, you see nothing, and then you start to see actually pretty nice self-assembly into these spherical objects as the hydrophobicity increases. So what we're doing is we're attaching hydrophobic molecules, and that's driving self-assembly. And so we wanted to use this strategy for delivery of chemotherapeutics. And so we attached doxorubicin. In fact, doxorubicin triggered self-assembly, and we attached paclitaxel and paclitaxel, both of them being reasonably hydrophobic. Paclitaxel being very hydrophobic, triggered self-assembly into very nice, near non-dispersed nanoparticles. The second thing I should mention is that the size is perfect for cancer. And again, we were lucky. It just so happens that these self-assemble into nanoparticles between 40 and 60 nanometers in size. And this size scale is critical because people have mapped the holes in the tumor vasculature. And the holes are sort of, typically less than 100 nanometers, sometimes they extend to a few hundred nanometers. So the size of the particle should be commensurate with the hole that it's trying to come out of, in a sense. If you're going to attach a drug, you have to attach it through some mechanism by which the drug can be released from the linker. This is very, very important because the ability of a polymer chain, for example, to get through to the nucleus, where some of these drugs act, is very limited. It's not going to go through the nuclear pore complex. This is well known. But small molecules can. So in our strategy is to take a drug, make a prodrug out of it, where this end, this malamide, would react with the cysteines. And here we have an internal hydrazone bond, which is well known to cleave very smoothly as the pH drops below 5.5. So when you drop the pH from 7.4, sort of physiological pH, into the late endosomal pH range, this bond should break and you should recover back free drug. And the hope was, once the free drug was cleaved in the endolysosomal compartment, it would then be free to diffuse, which was a question we didn't know the answer to. So we attached this, and you can see that at pH 7.4, there's no release. This is just a test tube. This is just changing the pH, looking at drug release from the nanoparticles. But when you lower the pH to 5, you get this very smooth release. You get about 75% release in 24 hours. The rest of it is probably still stuck on the column, we believe. Unfortunately, you can't see this, but what you see is if you take doxorubicin nanoparticles, which are red because they're fluorescent, and you incubate them with these tumor cells, you can see this on my computer, but not here, that about 30 minutes they get taken up, and you see red fluorescence in the nucleus. We know the polymer can get to the nucleus, and we know what's happening through other studies that we've done quite carefully is that the nanoparticle gets taken up, it ends up in the endosomal lysosomal pathway, and then somewhere along that pathway the drug is cleaved, the polymer stays in the compartment, the drug diffuses out, and in this case goes to the nucleus. So we do see the drug actually making its way in and then out of the endosome and then making its way to the nucleus, which gives it fairly good activity. So here we're looking at cell viability of tumors treated either cells in cell culture with nanoparticles or with the free drug. And you can see that the nanoparticle formulation just has slightly lower cytotoxicity, which is what you'd expect. When you attach things, when you take drugs, attach them to polymers, in vitro, you're often going to lose a little bit. But if you don't lose too much in vitro, you'll gain it all back and more, hopefully, in vivo, because the nanoparticles will stay in circulation much longer, and they will passively accumulate to much higher concentration than free drug, because the free drug is too small. Even if it's soluble, it's going to be excreted out very quickly. And indeed, this is what you see. There's no targeting ligands here. This is simply passive targeting. Just look at the data at 24 hours. Ignore the 2-hour transient accumulation. This is steady-state accumulation. Dox only accumulates to about 1% injected dose per gram. The nanoparticles accumulate threefold higher, 3% injected dose per gram. These are not impressive numbers, but the question really is, where is this threshold of therapy, right? So is it at 1 or is it between 1 and 3? And what we find is that even by going from this modest number of 1% injected dose per gram to 3, we see a profound effect when we do regression experiments. So here's an example of a syngeneic tumor, a colon carcinoma that mice get. We implanted it under the skin, either on the leg or the back. And then we did only one treatment. This is a very conservative treatment where the animals were treated either with PBS or doxorubicin at its maximum tolerated dose or the nanoparticles at their maximum tolerated dose. So this drug delivery system doesn't make a drug better. It simply makes it better tolerated and makes it accumulate better in a tumor. And what we find is that this is a pretty aggressive tumor. Animals start to die very quickly. Even a doxorubicin treatment, by 25 days, they're dead. And here you can see a clear visual example. This is at day 16. An animal treated with free drug has this enormous tumor growing out here. And you really can't see any tumor with the animals treated with the free drug. And if you look at sort of the punchline, it's not just shrinking tumors, it's survival that we care about. Treating patients, you can see the median survival is now 66. We typically get over 90% cures in this tumor model with a single injection. And this difference between the free drug and PBS is actually not statistically significant. So doxorubicin is not a very effective drug in this context, but you take this drug that is not very effective, simply package it into a nanoparticle through this very simple strategy, and it becomes far more effective. I should, however, confess that these are pretty easy tumors to cure, actually. Okay, so C26 is where you start. If you take dox liposomes, it works equally well. And Doxil, the dox liposomes are on the market. So we've made something that's probably as good as Doxil, maybe. If you really want to develop things, I think you have to continually challenge yourself with tougher and tougher tumor models. You simply cannot do subcutaneous models. You have to do tumor models, and you have to make them increasingly difficult, but you should start with an easy model, because if you fail there, then obviously you're not going to make it into the clinic. So we've challenged ourselves, and I'm going to show you data for 2 other tumors that are far more difficult to cure with doxorubicin. In fact, the first one, 4T1, is a breast cancer, which is almost completely resistant to doxorubicin. And you can see this here. If you treat with doxorubicin, all the animals, most of the animals die. If you treat them with a nanoparticle, you get much greater survivors. And here's Lewis lung carcinoma. Both of these are very aggressive, very metastatic tumors, and we can see a significant improvement when we treat with nanoparticles as compared to the free drug. So at this point now, we're looking at, for example, these are orthotopic tumors. These are implanted breast cancer tumors implanted in the mammary fat pad of female mice. So you're starting to recapitulate real disease, and you're working with a tumor that's, does not respond well to the free drug, and we're seeing significant improvement of the nanoparticles. So this does suggest to us that the system is somewhat robust, and we hope that as we take it forward, at some day, it'll help save the lives of patients. We've also looked at paclitaxel, and this is a triple-negative breast cancer. This is very difficult to cure. In fact, if you look at the survival of animals treated with just saline or with paclitaxel, it has absolutely no effect. So this tumor is completely resistant to paclitaxel, but if you formulate it as a nanoparticle, even at the same dose in this case, close to the same dose, 30 milligrams per kilogram, you can see a large increase in survival time. And then if you increase the dose to 50, you see long-term survivors. The reason you cannot use paclitaxel beyond 25 is that the animals will die because of neurotoxicity. So this is the maximum tolerated dose. So in this case, we are not only increasing the maximum tolerated dose, but we're actually somehow improving the potency of the drug, because even at very similar doses, there's a big difference between free drug and nanoparticle drug. And this is something that was a little bit unexpected to me. So at this point, all we've done is taken a hydrophilic polymer chain, attached a whole bunch, about 6 or 7 for doxorubicin, about 3 drug molecules for paclitaxel on one end. You attach these hydrophobic drugs, they surface some of the nanoparticles. They can take advantage of passive targeting and accumulate passively in tumors, and they seem to do reasonably well. At this point, we said we had many different options in trying to improve upon the performance. One is to take advantage of the fact that the extracellular pH in tumors is somewhat acidic. It's about 6.4, 6.8, and we could potentially use that as a trigger because we know how to build and disassemble nanoparticles in that pH range. Or we could simply put a targeting ligand. We can put antibody fragments on the outside on the corona of these nanoparticles, linear peptides, cyclic peptides, and so on. But the third and intriguing possibility that we decided to explore first was local hypothermia. And the idea is that you focus high-frequency ultrasound on a tumor and you heat it up for an hour or two to about 42 degrees Celsius, and we ask the question, could we now take these nanoparticles and turn on the heat such that only the nanoparticles in the tumor become really sticky, they become hydrophobic, and they aggregate together. And if they aggregate together over a period of an hour and we turn off the heat, we should have a very high local concentration, and we should really be able to drive the drug out through diffusion. So this is a non-trivial thing to do. To our knowledge, nobody has ever built nanoparticles that can do something useful in a 5 degrees Celsius range, between 37 and 42, inside the body. And we had to prove to ourselves we could see something happen in a living animal in a tumor. So we're going to use hyperthermia, and the idea now is these first-generation nanoparticles would just sort of, even in the presence of heat, they would remain hydrophilic, they would not aggregate, they just sort of go through the blood flow. And we wanted to take the system and make it very responsive to a 5 degrees Celsius change in temperature that is localized on the tumor, and we wanted to make them hydrophobic such that they aggregate and create a big clump, and then we want to turn off the heat and then dissolve them very quickly and create this large driving force for diffusion. So this was the challenge that Jonathan McDaniel was faced in his PhD, and it took him many, many years, and he developed this very nice empirical model where we can now predict the transition temperature of a nanoparticle, its aggregation temperature, as a function of both the chain length and the composition. So you see this sort of hyperplane here, and we can now pick out a region of interest. So all these polymers with this chain length and with this alanine content shown in red are capable of going through a phase transition according to his model between 37 and 42 degrees Celsius. So we picked a bunch of these and tested them. And to prove to ourselves that we could actually drive the phase transition, we have to actually be able to see the phase transition in vivo. And for this, we use a very nice system, a microscopy system that we learned from Mark Dewhurst many years ago called intravital microscopy, or the dorsal skin flap window chamber, where you take a mouse, you open up the skin flap, you implant some tumor cells, and then you close it up in this plexiglass chamber. And when the tumor grows to a certain size within this chamber, you're ready to do a microscopy. You take the animal, you anesthetize it, you put it on a confocal, cannulate the tail vein, and you can inject in the molecule of interest, and you should be able to see it within the tumor. And what you see here is an ELP labeled with a rhodamine, and this is coursing through the blood vessels of the tumor, and you can see them light up. And at the right magnification, you can actually go down to submicron resolution. We've done as well as 600-700 nanometers in the best case situation. So this would then allow us to actually visualize whether or not we could drive the phase transition of a nanoparticle, make it sticky, and make it aggregate in the tumor, thereby concentrating it there over a period of 1 or 2 hours. So I'm going to show you first the control experiment, which is the first generation nanoparticle, which doesn't go through a phase transition, and you will see really nothing because these are nanoparticles flowing past, so they're below the diffraction limit. You won't see anything happen. And this movie is running, and you really can't see anything. You might see a little bit of motion artifacts. It's still running. And now I'm going to show you what happens with the second generation nanoparticle. We designed something where the nanoparticle should become hydrophobic sticky, aggregate in the tumor, and it was actually quite dramatic. It took a long time to get it to work. We had to figure out a lot of details in the design. Sorry. There we go. So these are nanoparticles that are flowing past, but they're no longer nanoparticles. They've aggregated. Here you can see that we've actually managed to block off a whole blood vessel. It's almost like lava flowing slowly. This whole thing is sort of big, viscous goo of nanoparticles aggregated together. And so you can see we have a very high local concentration in the tumor, but this is under heat and they're aggregated. They can't really diffuse very far. And the idea, the million-dollar question is, what happens when you turn off the heat? We know we're going to have a very high local concentration. Will there be an impact? And that's what we're working on now. So I just wanted to sort of end this part of the talk and sort of summarize it, that we've taken these single segment, very simple ELPs, simply put attachment sites at the end. And I should point out, even building this simple design is not so easy to do with synthetic polymers, where you have only 8 reactive sites on only one end of the polymer chain. We attached doxorubicin. We showed that doxorubicin and paclitaxel both create nanoparticles that seem to be quite promising. And now we managed to design, or Jonathan has managed to design, a second generation nanoparticle that shows this large transient accumulation in the tumor. And the next step, really, we think the clever thing to do would be to take it and combine it with vascular targeting, because these things are sitting right next to the endothelial cells. We want to put vascular targeting ligand and hope that they will be endocytosed. They'll be taken up by penocytosis into the and receptor-mediated endocytosis into the tumor, into the endothelial cells of the tumor. But we don't have those results yet. Next, I want to switch gears and talk about peptide drugs. The peptide drugs are interesting. There are many of them in development because they're highly specific. They can have very high potency, nanomolar or better for the receptors or targets. They're generally not very toxic, but they also have a lot of problems. And one of the biggest problems is that they're peptides, that they're short. Even if they're hydrophilic, they get very clear. The half-life of a peptide can be as low as 2 minutes. In fact, the example I'll talk about today, its half-life is 2 minutes. You inject it, it's gone. Half of it is gone in 2 minutes. And that's not very good if you're trying to deliver it in the context of treating a disease. So, people have been working on this problem for a long time, and there are many, many different strategies, and I'm not going to go through all of them. I'm going to talk about some of the more bioengineering type strategies as opposed to the more molecular strategies like fusions and so on. So you can take the peptide drug, you can put it in your favorite degradable polymer like polylactic glycolic acid, and there are hundreds of papers published on this, make a microsphere, and it has some solubility. You can take the microsphere suspension, inject it under the skin, the microspheres sit there because they're pH sensitive, they will hydrolyze, and then release drug. This is a very popular strategy. It's actually used in the clinic. The drug I'll talk about today was recently formulated as a PLGA microsphere, and it's now in the clinic. It's called Bidurion. You can also put things into liposomes and micelles. You can create transdermal patches. You can create pumps. So these sort of engineering solutions. But all of these, I would say, the microspheres are sort of not the gold standard, but they are very popular in terms of creating sustained release systems for drugs, especially for peptides. The challenge we posed ourselves was not that we don't like microspheres and we don't like PLGA, but we said, we work with these kinds of genetically encoded polymers. Can we make peptide polymers? Can we simply write a sequence out that will recapitulate everything that a microsphere does, but gets rid of the microspheres? That was the challenge we set ourselves. So we wanted an injectable polypeptide sequence, something that was soluble, but would create a depot upon injection under the skin, and would show sustained release of active peptide drug, where we could control the kinetics, we could tune the kinetics of release, but not use these things, not use these PLGA microspheres. So a PLGA microsphere system without the PLGA, and to do it all by writing a sequence out. What happened here? So we call this system protease-operated depots. This is an injectable polypeptide that does mimic the microspheres. And how would you do it? So we're very simple-minded. We said, let's just write out a sequence which has two things. One segment is going to be the segment that will make the depot. The other segment will be the drug. So we made a diblock copolymer where one segment is an elastomeric polypeptide with a very simple property that its LCST, its transition temperature, is between room temperature and body temperature, between 25 and 37. This would be the depot-forming module or segment, if you will. And then we need to deliver quite a bit of drug. We want to be able to control the loading of the drug. So we simply said, let us simply take the peptide drug, hook it up, and make another segment, make a polymer of the drug, with a protease cleavable site. So just put in a site that an enzyme, some unspecified enzyme under the skin will attack. We didn't know what to make of that, so we put in arginines, which are attacked by a whole bunch of proteases. This system does recapitulate on paper some of the features of microsphere delivery technology because we can tune, for example, loading of the drug simply by controlling the block ratios. That's the simple way of controlling drug loading. And the idea would be that you would take this soluble polymer chain, it would be in a syringe, you would inject it into the skin, body temperature, body heat would drive the phase transition, the soluble polymer would go through its phase transition and become this viscous coacervate, so this viscous goo under the skin. And the question was, would it sit there long enough? And if it sat there long enough, would proteases actually come in, attack it, and liberate not oligomers, but monomers? Because it turns out for many drugs, even when the oligomeric peptide drugs don't work, and the drug I will talk about today certainly doesn't. It has to be a monomeric drug because its activity is located at its N-terminus. So what drug to pick? There are 650 peptides we could have picked, but we picked glucagon-like peptide because it's a drug that's already in the clinic. But GLP-1 is the master regulator of insulin production. So when you eat, the intestinal L cells, in response to eating, will produce GLP-1, which will then make its way through the bloodstream to the pancreas and activate the GLP-1, bind to the GLP-1 receptor, and then that leads to insulin production. The problem with GLP-1 is that it has a half-life of 2 minutes. It's evolved to have a half-life of 2 minutes, so it's not a very good drug by itself. Despite that, people have come up with, or companies have come up with variants, analogs, mutants. In fact, Byetta is a drug which comes from the Gila monster. So the Gila monster also makes its own GLP-1, oddly enough, and that works for twice a day because it's a little more protease resistant. And then there is an acylated version which hops on, binds on to human serum albumin, which is approved for once daily injection. And very recently, a once weekly came out that's called Bidurion. That's a PLGA microsphere, but it's not very popular because the drug is actually far inferior to this one here, pharmacologically. So the question was, could we create a protease-operated depot of GLP-1? It turns out that when you do these things, you never know if E. coli is going to like it or reject it. In this case, it liked our system, and so here you can see we can make lots and lots of the pod, of the protease-operated depot, from this SDS page gel. We can purify it to homogeneity. In fact, we can get about 150 milligrams per liter. We can tune the loading of the drug, so we can have a polymer chain. We can either put 3 repeats, 4 repeats, 6 repeats, or 8 repeats of the drug onto the polymer at the gene level. So this way we can tune it by more than a factor of 2. And we can make systems where you can tune the transition temperature. So here are 3 different pods all attached to an elastin-like polypeptide with 3 different molecular weights. So not only can we tune the transition temperature, we can also tune the loading. And here's a soluble control where the GLP-1 polymer or oligomer was attached to a ELP that had a much higher transition temperature. So this would be sort of the control for depot formation. What we see when we do cell culture studies where we look at receptor activation of the GLP-1 receptor in cells, is that GLP-1 is a very potent drug, sort of nanomolar potency. This is just the free peptide. However, if you incubate the cells which are overexpressing this receptor with the polymer, it has no activity. So the depot by itself is completely inert. It has no activity, so even if it would release all of it, nothing would happen. This is because the fusion has no activity, because all the activity is localized in a monomer unit, because it has a real steric requirement for the N-terminus to be available to the receptor. So if you add a protease and start chewing it up, what we see is that in a matter of hours, we can recover the activity very cleanly. And this is because of release of monomer by the action of protease. So the system can be triggered by the action of proteases releasing monomer that acts on the receptor. We next went to do some animal studies with pre-diabetic mice. If you inject even PBS, these mice get stressed very quickly. You can see that their glucose level goes shooting up just because of stress. So this is not an artifact, is that if you inject just PBS or just handle them, they get really stressed. If you inject GLP-1, just the peptide by itself, you can get control for 2 hours. That's it. All you get is 2 hours of control, which is consistent with sort of 12 hours in humans, because mice are metabolically more active. If I now take a control system, an inactive pod, where the peptide drug, the oligomers of the peptide drug, were attached to an ELP, elastin-like polypeptide, but the elastin-like polypeptide had a transition temperature much higher than 37. So it's soluble in the syringe, soluble under the skin, so it really doesn't make a depot. It just sits there, and then it dissolves and diffuses. We get a little bit of better control simply because we have a longer molecule. So it gets cleared less quickly, but we get control of glucose for a day. This scale is now 144 hours. Here it was only 4 hours, okay? If we next make a pod where it's soluble in the syringe, but makes a depot under the skin, we actually imaged it, and we used fluorescence tomography to show that even at 120 hours, we could see a residual depot under the skin. And the consequence of that is that we can get control of glucose levels for up to 5 days. So from a single injection of this very simple system, we can control glucose levels in mice for up to almost a week without the use of any external excipients like PLJ microspheres. So just to summarize this part of the talk, a pod is nothing but a modular injected depot that consists of two components, a depot of a thermally responsive coacervate forming polymer, so it goes to the phase transition, makes a depot, fused to multiple copies of a peptide drug, which is GLP-1 in this case. It's soluble at room temperature, but when you inject it in, it makes a viscous coacervate under the skin. Unknown proteases at this point, we don't know which proteases they are, come in and actually liberate enough free monomeric drug that we can see control for up to 5 days in mice. We can also tune the release kinetics, which I didn't talk about, by changing the design of the ELP. As we change the transition temperature of the ELP relative to body temperature, we can make depots that are stronger or weaker. So we can make things that last 1 day, 3 days, and 5 days. Where do we go from here? I want to sort of end on a more philosophical note, because everything I've talked about today has been really based on this one very boring sequence, ELPs, VPG, XG. We've been trapped in this karmic cycle for my whole career. I started and I said, I'll work on ELP 5 years, get tenure, move on. And here I am 17 years later, making the same polymer over and over again. You feel like a gerbil on a wheel, right? So why? Because the obvious realization is, if you look at, we have 20 amino acids, we can string them up in infinite number of ways. There aren't enough atoms in the universe to make the polymers we'd like to make. In fact, Felipe, whose work I'm going to talk about next, Felipe Garcia-Quiroz, sort of did this calculation of 100 amino acids, you have 10 to the 130 possibilities. What we've been doing, if you look at this, Felipe's representation of sequence space is very simple. We've been sitting here in one spot in sequence space called valine, proline, glycine, X-glycine. And so X is like our only variable we vary, and we make these polymers, and they're useful, no doubt, but it's a bit boring in terms of material science. And we've been sitting there because we are sort of obsessed with this LCST behavior, where you raise the temperature, things go from one phase to two phase, which are very useful. We recently started to do the opposite, look at UCST, but I have no time for that today. So this is what we've been doing. This whole talk really has been, in some ways, playing with this polymer sequence, turning on the transition temperature, turning on the transition, turning off the transition, and coming up with some sort of bioengineering and medical applications. The question was, what would happen if we went across sequence space? Would we see other interesting properties? Would we see new polymers that have different kinds of LCST behavior, UCST behavior? So if you are trained in sort of biochemistry, as I was during my postdoc, if you give somebody a protein in biochemistry or protein engineering, say, tell me more about it, they'll give it to a graduate student and say, do mutagenesis. Map out the active site. Start changing all the amino acids in the active site of the protein or enzyme. And we said, this is a system that's 5 amino acids long, but nobody has ever done mutagenesis on it. And this is so simple to make mutations with. The problem is that if you want to sort of look at even 5 amino acids and make all the different variants, they end up being quite a few polymers. And up to this point, it was to take us weeks to months to make new sequences, not because making the sequence is difficult. It's the assembly that was very tedious, because we take monomer, make a dimer, tetramer, and so on. Take weeks to make a library. So I sort of had this crazy idea of how to do this. Of course, my ideas are usually half-baked, so it took a very smart student, Mira, and Felipe working together to take these half-baked ideas and make them work. My idea was, why don't you circularize it and then spool sausage off it and just get a long polymer one. It sort of works, but doesn't quite work. There were other tricks involved. And so Mira and Felipe invented this thing called overlap extension rolling circle amplification, where you take a single-stranded DNA, make a circle. Then when you have a polymerase that spools around, it's not a very good polymerase, so it just makes short oligomers. But those short oligomers are self-complementary because it's a repetitive sequence. So they bind to each other. Then you can do polymerase chain reaction with primers at the end. And what you end up getting from a single tube is a smear, which doesn't look very good. It's basically what you have is the same polymer, same composition, but this is a DNA, this is a gene that encodes for many, many different repeats, all mixed together. But the beauty of cloning is separation in biology. When you clone it, each gene only goes into one cell, which is sitting on a dish, and now you can recover back genes that encode for discrete molecular weights. So you can see here, these are DNA isolated from different colonies, and now we have very clear bands corresponding to a gene that encodes for precise molecular weight. And then when we put each of these genes into E. coli, you make these polymers. So in a 96-well plate, I can end up with 96 different genes that I've made with n number of molecular weights encoded. So this is a very powerful and quick method of doing sort of saturation mutagenesis. And the question will happen if you simply put an alanine at every position. It's called alanine scanning. It's done in biochemistry all the time. So here are all the different genes. Mira and Felipe didn't even bother making all of them. So the white are all the genes they isolated. The green are the proteins they made. And what was really surprising is all of them had phase transition behavior. This turned out to be completely promiscuous. We did not expect that you put an alanine, you insert an alanine or substitute an alanine at every position, you get phase transition behavior, with one exception which I won't talk about. But 12 out of 13 polymers had phase transition behavior, which suggested that we were sitting on this point, sort of very scared to move around, to take excursions. When we walked, the terrain was very friendly. We had phase transition behavior. So the question was, how far could we venture out on this hike, and what would we find? So at this point, Felipe decided to just go back to basics and look at sequence space more broadly by doing some bioinformatics studies. So what he went back to was to look at elastin and many other intrinsically disordered proteins because they share sort of some very interesting phase behavior properties. If you look at elastin across different species and you look at the canonical motif, VPGXG, it actually doesn't occur that often. But tropoelastin, all elastins have the same phase transition behavior. But the sequence, the putative canonical sequence that's supposed to give it this behavior is actually pretty uncommon. So Felipe said, you know, what is common is P and G. Let me just relax the sequence constraints. Say, how often does PG repeat? How often does PXG repeat? PXYG, all the way up to 4 residues between P's and G's. And when you do that, tropoelastin studded with P's and G's variable distances apart. And so his hypothesis was it's simply P's and G's, a certain distance apart, that drive phase transition, because the hypothesis was you need an unstructured system to see phase transition behavior. So he went and very ambitiously made a huge number of polymers. So P and G, nothing in between, P and G, some residue in between, and so on. And what he found was shocking, or maybe not so shocking at this point. Everything had phase transition behavior. Look at this. There's a lot of detail embedded here. I don't want to take you through all the details. Just want to show you. All of them are going soluble to insoluble. Soluble, the transition temperatures change and so on. But the beauty was now that we had such a large set of 50, 60 polymers, we could now look at them and do sort of simple pattern recognition. We could start classifying things according to the behaviors. And one thing that he found was very interesting was that he could classify these polymers into 3 classes based on the hysteresis and the phase behavior, meaning that when you go up, you get one transition temperature. When you come down, if you have no hysteresis, you get the same transition temperature. If you have hysteresis, it'll be different. And he found 3 classes of behaviors. Systems that have no hysteresis, so you can see almost no hysteresis, the ones in red, which is the canonical elastin sequence. A sequence that has a finite, significant hysteresis, but it's finite. And then systems where you go up and you come back, and these things don't come back, if at all, ever. This is not aggregation. It's still a phase transition behavior, and I can explain that if you're interested later. The question is, so we have this hysteretic behavior. What is it useful for as a bioengineer? This is just sort of biophysics. You could say, well, this is what biophysicists do. We need to do something useful with it. This is useful and important because if you were to take, for example, a diblock polymer where one block has LCST behavior and you heat it up, it forms a micelle. Fine. But you turn off the temperature, it's going to dissolve instantaneously, which could be fine for certain applications, but in other applications, we'd like to use heat to drive the formation of nanoparticles in vivo at a site in the body, turn off the heat, and have them be stable, retain their morphology, right? We want to encode sort of a memory effect in there. If you use a system that's highly hysteretic, we think we should be able to use kinetics to trap that morphology. So in other words, use a block that makes the core of the micelle to have a high degree of hysteresis so that once it forms, it can simply not come back on a relevant time scale and the nanoparticle will remain trapped. And so Felipe actually proved this. What you see here is 2 things. We're just looking at turbidity on this axis, light scattering on this axis. So if you look at the turbidity, you go up, you hit the critical micellization temperature, the turbidity goes up, and then it's stable in this range. If you do light scattering, you can see no nanoparticles, and then nanoparticles with a radius of about 50 nanometers, so diameter of 100 nanometers. And then when you lower the temperature, we're still doing the DLS on the bottom end. We haven't done that yet. But if you lower the temperature, you see that these things are stable. So what we've done is at around 39, 40 degrees Celsius, we form nanoparticles. And now when we cool, they're stable because the turbidity doesn't come down. So this is a way of using hysteresis to create kinetically trapped structures, which would have some applications in medicine. The fact that we now have this very expanded vocabulary, that it's not just PG, it can be P's and G's separated by variable numbers of promiscuous residues, also allows us to ask the question, why is it when people make delivery systems, they always have a delivery part and a drug part? Can we take the 2 and fuse it? Can we take things that are stimulus responsive and embed in it within the sequence, do almost like an in-frame fusion, if you will, of another activity? So this is what Felipe set out to do, and he made 2 different polymers. This is GX blah, blah, blah. The second sequence is simply the X transposed and moved here beyond the P. So they have the same hydrophobicity, same composition, just minor differences in sequence. And he made them with a cysteine at this end, with the repetitive cysteine, so he could actually make a hydrogel in vivo and injected it into mice. Interestingly, in this case, the transposition has no effect on the LCST because LCST is largely controlled in this case. The temperature is controlled by the overall composition hydrophobicity. So these polymers are not distinct at all in terms of their physical behavior, in terms of how they go through the phase transition. But if you take these two polymers, mix them with tumor cells, and do a tumor engraftment experiment, we ask the question, does this have any activity? We saw radical differences. So if you just do PBS, the tumors grow pretty quickly. This is at some time point. He measured the tumor size about at this size range. SM24, this one here, has no activity because it does not prevent engraftment of tumors, and this polymer, SM1, has some intrinsic bioactivity that prevents tumor engraftment. We have no idea what the actual activity is, but this is an example to show that we can now start to embed bioactivity within the polymer sequence that still retains its phase transition behavior. So this is interesting to us philosophically, because if you think about the current state of the art in designing polymers, designing systems, we always have this sort of separation of the two components. We have a physical property part and a biological property part. You have a micelle with a ligand, with a peptide ligand, or you have an antibody with a polymer. What we'd like to do in the future is to be able to take these two and seamlessly fuse them such that you have multiple properties all encoded in one seamless system. So just to sort of end this part of the talk, I'd like to sort of summarize and say that this exploration of sequence spaces, hiking, has been very, very productive and useful. We have discovered a very large set of sequences that have LCST behavior. These have different properties. They have different levels of thermal hysteresis, which is very useful for biomedical engineering. And it allows us to create structures, for example, with tunable self-assembly. We can now start to seamlessly embed bioactivity within physical properties. And we think that hopefully this points a way where the world of material science can be fused with the world of pharmaceutical science. And that's sort of, it's a very broad ambition and a hope and a dream. But this is where we'd like to push our research in my lab in the future. With that, I'd like to thank all the people who did the work. I've been very lucky. I've had extraordinary students and some very good postdocs the last 17 years. The ones in bold are the ones whose data I showed specifically today, though I have to give credit to everybody in my group. This is the group as of a few years ago. Here's Felipe, here's John, whose name came up multiple times. But there are other people here who also contributed. Sources of funding, I'd like to thank Dan Hammer for solving the dilemma of doing cryo-EM. We could never do it. Now we finally learned. And National Institutes of Health, without which none of this would be possible. My group here, I'd like to thank them once again, and you, the audience, for listening patiently as I rambled on. Thank you very much.
We have time for questions for Dr. Chilkoti. There's microphones strategically placed. If you have a question, if you could address a microphone, we'd appreciate it, because we're recording this session.
So that was a very fantastic talk and fantastic work. I wanted to know if you can please comment on the immunogenicity of these peptides.
It's a very good question and a fair question. Immunogenicity, right? So peptides can be immunogenic. The answer is it's gone into about 600 patients. There's been almost no antibody response. So depending on the sequence, they can be poorly immunogenic or be highly immunogenic. I think we were lucky. We made things that were very boring biologically. They had, at the guest residue position, they had glycine, alanine. Look at the side chain. There's very little prospects of molecular recognition. And ultimately, all of life is nothing but a game of molecular recognition. So if I start putting in hydrophobic amino acids, I bet I can make really immunogenic things. So if you're lucky and you can be non-immunogenic, but if you want to make immunogenic things, you probably could. I think I have.
So you mentioned that when you have the same drug amount, but when you use the nanoparticles, they become much more effective. What is the basic mechanism that when you have nanoparticles, they become more effective?
So in the context of cancer, it's sort of simple. We're just using the leakiness of the blood vessels. We're making nanoparticles that are very soluble, that are very hydrophilic. They're not being cleared very quickly through the kidney, so they stay in circulation. Because they stay in circulation with every pass as they go through, they have the opportunity to diffuse through the leaky tumor vasculature. Then they go and sit there in the stroma, then they fall apart, polymer chains diffuse, they get taken up. It's just a matter of increasing the concentration by taking advantage of the leakiness. The drug molecule, even if it was hydrophilic, would be cleared. And in a worst case like paclitaxel, it's completely brick dust. So it's a matter of creating a high enough concentration long enough to take advantage of the leaky pipe effect, which is what the EPR effect is.
Question over here.
I was interested in knowing what's the size of the polypeptide that you can put between repeat sequences and still keep the phase transition behavior. Because one could think about putting like whole protein in between and have this
Oh,
gel-like
so
behavior.
you're asking how robust is this, how tolerant is the system to insertions and fusions?
Yes.
So I didn't talk about it today, but we make fusions all the time. We take a protein, we fuse an ELP, and every soluble protein with a soluble ELP goes through phase transition behavior. The details matter. So highly charged proteins will drive the transition temperature up, somewhat hydrophobic proteins will lower it. So the system can take very large fusions and still have phase transition behavior, but the fused partner will affect the transition temperature. So you have to engineer for that or compensate for it. And you can certainly take an ELP and put a protein in the middle and it'll still have phase transition behavior. I mean, the system is very, very tolerant.
Anyone else? Well, let's thank Dr. Chilkoti again. Thank you very much. The celebration of Minorities Luncheon is at 12 o'clock, and we're back in here at 5:45 for the BMES Annual Awards Ceremony and President's State of Society Address. Hope to see you then.